Hello, so today I'm going to talk about lysogenic conversion and lytic and lysogenic replication. Lysogenic conversion describes a situation in which a bacterial host is able to acquire a new trait that is a direct result of the expression of a gene encoded by a lysogen. This can change the phenotype to a pathogen. There are a number of stages involved in this outcome, first involving lytic and lysogenic replication. The lytic replication cycle is a 5-step process. The first step involves attachment. Here, the virion finds a host cell and attaches to it. Next, the virion enters into the host. After that, new nucleic acids and viral proteins are synthesized by the host cell using its ribosomes and enzymes. Next, new virions assemble inside the host. Finally, more new virions are then released from the host cell, and the cycle repeats. The lysogenic replication process has a modified cycle where the infected hosts are able to grow and reproduce normally up until the time that they lyse. In this cycle, phages are called either temperate phages or lysogenic phages. First, the virion is in contact with an E. coli cell, then attaches to it by the tail. The DNA of the virus enters the cell, but in this process, DNA is not destroyed inside the host cell. The virus just remains inactive at this time. This is called a prophage. This prophage stays inactive using a protein that is able to suppress prophage genes, but as a side effect, this protein also causes the bacteria to be resistant to any further infection if the other viruses are of the same type. Also different from the lytic cycle, the prophage becomes a physical part of the chromosome of the bacteria by inserting itself into the DNA. All it takes is the fusing of 2 pieces of DNA. One piece of the virus's DNA fuses to the cell chromosome. When the chromosome replicates, so will the prophage. This would mean that the infection will spread to all of the daughter cells of this lysogenic cell. It is still considered inactive at this point. If the prophage is removed from the chromosome, whether by recombination or another method, it will then re-enter the lytic phase. This is called induction. These inductive agents resemble agents that are damaging to DNA molecules as they have the same physical and chemical components. Some examples of these include X-rays or carcinogenic chemicals. Once induction has occurred, the steps of synthesis, assembly, and release that we see in the lytic cycle now occur. This causes the cell to break open because it is filled with virions. Basically, what has happened is the phage inserts specific characteristics into the bacterial genes, causing the bacteria to have better survival. 2 examples of when lysogenic conversion has contributed to pathogenicity is in the diphtheria exotoxin of the bacterium Corynebacterium diphtheriae, and the streptococcal purogenic exotoxin produced by rare invasive strains and scarlet fever strains of Streptococcus pyogenes.